Hi again. Today I'm going to be talking about a very special topic, a biochemical topic on storage disorders. These are rare monogenic metabolic disorders. Um, they are so-called inborn errors of metabolism. And I'm going to convince you why these inborn errors of metabolism help us to better understand what tissues there are in our body, about the cell cycle of these tissues, and also about the basic biochemistry of your cells. So let's begin. So this video will be split into a few segments. The first is going to focus on lysosomal storage disorders. Then I'm going to focus on mucopolysaccharidoses, and lastly on glycogen storage disorders. This is just an introduction to the biochemical principles of these conditions, and I will not be focusing on extensive pathology or patient symptomatology or even treatment. So first of all, lysosomal storage disorder. Let's break it down. What is a lysosome? A lysosome is an organelle within the cell that contains lysozymes, which are enzymes that are designed to break down or degrade substances in the cell. Usually there's an ingestion of a material into the lysosome, followed by the release of activated lysozyme. In lysosomal storage disorders, there's a loss of lysozymal function, and this can happen for many reasons. It can happen because the enzyme is defective, or because the cell membrane of the lysosome is defective. Many reasons. Genetically, nearly all of them are autosomal recessive. Now, what is this thing about autosomal recessive conditions? They generally tend to, so you require a homozygous genotype to have the phenotype, and there's usually a near complete penetrance if you are homozygous. You can be a heterozygote carrier with some symptoms, but the real disorder happens in a homozygous state. They usually present very early, so usually it's a pediatric issue, sometimes a neonatal issue. And what is the pathological problem here? Well, what I like to think is that our body we ingest usually relatively simple carbohydrates, proteins, and lipids, but our body synthesizes really complex and insoluble substrates, and these can include complex glycoproteins, which play a role in receptors. For example, if you look at my platelet cell biology video, I talk about 2 glycoprotein receptors that are important in thrombosis. Also glycolipids, which are very important for cellular signaling. For example, not glycolipids, but phospholipids are particularly very important, for example, in the arachidonic acid pathway. Yes, mucopolysaccharides. So what is a mucopolysaccharide? It's basically a polysaccharide with a mucus type functionality or physics. So a polysaccharide is obviously a sugar, a complex sugar, and the muco bit means that it's more on the sticky side, so it's to do with connective tissue. And lastly, glycogen. So glycogen is a large carbohydrate from, as I believe, alpha glucose. So alpha glucose forms the 1,6 glycosidic bonds in a chain, and then there are side chains to form your glycogen molecule. So these are all pretty big molecules. They're usually insoluble, and they need to be carefully trafficked in the cell via the cytoskeletal structures like dynein arms. Lysosomes play a role in breaking down these substrates when there's too many of them, or when they are on some level defective. Let's begin with the first set of storage disorders, Tay-Sachs disease, which is an example of a gangliosidosis. What is a ganglioside? It is an example of a glycoprotein. In Tay-Sachs disease, you have failure to break down a specific glycoprotein called ganglioside M2 or GM2, and this is due to a deficiency in an enzyme called hexose, which is hexose is 6-sugar, aminidase. So think of it, hexose is like the sugar bit, aminidase, amine is like the protein bit, and ase is the enzyme bit. Hexose aminidase. And this is due to autosomal recessive homozygous mutations in the gene encoding this enzyme. So what is the pathologic consequence of this? Well then, these lysosomes cannot degrade this thick glycoprotein molecule, GM2, so it builds up inside the lysosome, thickening it. This leads to the lysosome having a whorled appearance, a bit like an onion, and you can see this on histology. Now, I mentioned in a previous video about how when we produce proteins, we have chaperone molecules that help to regulate if it's misfolded or not. So glycoproteins, if they build up too much, our chaperones will help, try to degrade them, or there will be changes in gene regulation so that these protein synthesis is down-regulated. Now, if too much GM2 builds up in the lysosome, this is a stress, and it triggers the apoptotic pathway. Now, which cells in the body will this be affected the most? I'll give you a moment to think about it. Okay, so let's see if you got this right. It's going to be cells which are in the quiescent stage and are non-labile cells. So it's not going to be hepatic. It's definitely not going to be hematopoietic. It's going to be something like neuronal, or you could have said cardiac. But in Tay-Sachs, you have rapid loss of neurons very early on in life, which can lead to developmental delay, mental retardation, and so on and so forth, and even neurological deficit. Moving on, we have Niemann-Pick A and B. This is now a disorder of glycolipids. Okay, so glyco is a sugar, lipid is lipid. And this is due to the lack of an enzyme called basically sphingolipidase. So enzyme that breaks down sphingol is like sphinx, is just a Greek, Egyptian thing, a Roman thing, I guess. Sphinx, sphingolipid. So think of it as like a very complex lipid. Now, the distribution of this pathology is different to Tay-Sachs because it occurs in macrophages. And so you have development of these accumulation of sphingolipids in macrophages. Now, if you understand where macrophages are found in the body, you will appreciate where the problems can happen in Niemann-Pick. Namely, the spleen, which is where macrophages dwell as a secondary lymphoid tissue, the liver, because you have your Kupffer cells and also general antigen presentation, and also your bone marrow, because that's where your macrophages come from the hematopoietic lineage, and also obviously your lymph nodes, which is where macrophages reside. Furthermore, you can also get neuronal damage because, as I mentioned before, if you have accumulation of lipids in neurons, which can't regenerate, you have neuronal loss. Type C Niemann-Pick is different, and I'm not going to go into that today. Now I'm going to move on to Gaucher disease. So Gaucher disease is very similar to Niemann-Pick in the sense that, so you begin with your sphingolipid, which should be broken down into a downstream molecule called ceramide. So in Gaucher disease, you cannot break down ceramide because of a lack of an enzyme called glucocerebrosidase. And again, the cells involved are generally macrophages and phagocytes, which have accumulated levels of ceramide, like toxic lipids in your cells. However, in Gaucher disease, the pathology is much more immunological because these phagocytes then release cytokines which increase osteoclast activity via the rank ligand, leading to bone resorption and osteopathy, and also, as I mentioned, as in the previous condition, splenomegaly, because that's where phagocytes and macrophages accumulate, and also neurotoxicity because it's a non-regenerative cell. So we've talked about the storage disorders. I'm now going to focus on the mucopolysaccharidoses. So in fact, I've decided actually to split this video into two for ease of your comfort. So in the next video, I'll be talking about mucopolysaccharidoses and also the glycogen storage disorders. See you in the next one.